All right. Hello, this is the recorded version of lab today. Maybe it won't be the best recording, but you get some of the experience of being here live. All right, so we had that one lab class where we talked about experimenting, and I thought it'd be nice to get some practice, set up an experiment, and hopefully it works out okay. If the experiment doesn't work okay, at least we'll learn something about some plants. So today's plant of interest is a floating aquatic fern called Azolla. So it is a fern. It does reproduce with spores. It belongs to a group of aquatic ferns. It's a relatively rare growth form for any kind of plant, let alone ferns. And I chose this one because we have a lot of it in the greenhouse. It grows really well. And it has an association with a Cyanobacterium. So it's sort of like a condensed stem. I think there's stem in there and leaves. And then on the handout, it shows you how like a cross section through one of these leaves here. And this plant has a special association with Cyanobacteria. So it's like the there's a special compartment inside of the fronds on those floating leaves. And that's meant to house Cyanobacteria. And we talked about this when we talked about Cyanobacteria. There are some symbiotic Cyanobacteria. This is a fern that can still do photosynthesis, so it doesn't need to partner with the Cyanobacterium to do photosynthesis. But Cyanobacteria have an ability to do nitrogen fixation. So inside of that cavity, we'll give it a look. And hopefully we can crush open some of these fronds and see the Cyanobacteria inside. They're going to look like kind of a string of beads. And every once in a while on there is a cell called a heterocyst, which basically means a different cell. And that's a cell that's specialized to do nitrogen fixation. So what benefit do you get by partnering with nitrogen fixing bacteria? Better have. Nitrogen. All right, nitrogen. So plants need nitrogen. This is one of their essential soil nutrients. If you buy a fertilizer, nitrogen is really the first piece of that ingredients that they call attention to. So they say most fertilizers have nitrogen, potassium, phosphorus, and nitrogen is a big one. Important for making proteins and DNA and chlorophyll. And so if you are a plant and you're growing in, this is a plant that grows just in the water. If you're growing in water that's a little low on nitrogen, this is something you can partner with this cyanobacteria and actually makes nitrogen available for you. So this kind of raises interesting questions about sort of the fertilizer availability of nitrogen to our Azolla ferns. So, what I've got is basically a setup we can turn into an experiment. I left it a little open-ended on the handout, but I think we'll probably all just come around to doing the same thing. But we can basically set this up, kind of walk through the process together and plan an experiment for finding out something. So I have in my mind what I'd like us to do, but I think I'll try to get you to the same conclusion. So I have different kinds of water, and they're all based on tap water, so we're not doing a sterile environment. Trying to get things sterile, I think, is a little bit extra layer of difficulty for us that we don't need to worry about. I don't think there'll be big contamination for anything. I guess we'll solve that if we get there. And the tap water will be a little more balanced osmotically for just nutrients, background salts, and so on, as opposed to deionized water where we might have to add all the nutrients we need to get that up to speed. I do have water that we added some extra nutrients to. So there's one that I'm abbreviating MS. So on page 2, I mentioned the ingredients we have. I've got 4 flasks here, each with a different kind of water. So the MS is abbreviation for Marashika and Skog, one of the older publications where people figured out, basically early on, if you wanted to grow a plant, or algae or something like that, if you wanted to grow it in a chemically defined environment, we talked about that a little bit. This is basically the set of nutrients that are the minimum that plants need. Not just the minimum, but like an essential array of nutrients that a plant would grow well with. So it has their nitrogen needs, their phosphorus needs, their potassium needs, and then even some trace elements, magnesium, calcium, all the way down to, I don't even know what the finer, manganese is a little bit in there, I think. So if the plants need more nutrients than they can get from tap water, we might see a difference in how well they grow if we add nutrients. Another addition is the addition of ampicillin, abbreviated AMP, and this is an antibacterial. I don't have any penicillin allergies or anything new. Cool. Took a gamble on that one. Let's hope we don't find any today. So, what in the world would this do for us? Control the bacteria? All right, so my hypothesis here is this might have an effect on the abundance of the cyanobacteria. So with that addition, I'm hoping to actually kill out the cyanobacteria, or maybe at least inhibit them a little bit. So we have basically the addition of outside nutrients, and then the antibacterial or antibiotic chemical might knock down the plant's ability to make its own nutrients. So, an early hypothesis, if we see a difference in how well these grow based on the availability of nutrients, adding nutrients might cause them to grow better, and then removing their ability to get their own nitrogen through the cyanobacteria, that might affect their growth more negatively. And who knows? Maybe we'll see an effect of adding nutrients and taking away the cyanobacteria. So I guess early on, if we want to set up an experiment where we are farming the native cyanobacteria, probably make sure they're in there. Anybody have thoughts so far? That's pretty good. All right, so I've brushed up these successfully. Ta-da! Show my computer screens to each other. All right. So this little bit down here, these are the cyanobacteria. And I might try to find some better looking ones. That kind of looks like a heterocyst to me. But it being on the end of this, I'm not quite sure. All right, I'm going to maybe leave it at that for now. Yeah, that looks a lot like a heterocyst. So bigger, rounder. You can also see, oh, so cute. So we're definitely not a sterile environment. There's a little like euglenid or something in there. These little jiggly bits in there would be organisms that are associated and kind of swimming around in the medium. So we're gonna sort of take that as fine and hope to change or see an effect based on what we change and not an effect based on all that. So I guess that's a nice entry into the experimental part of this. So So how should we set up an experiment? So I guess regardless of what we expect or whether we see an effect, we should talk about how we set up an experiment to be able to see an effect. So what kind of things are we going to hold constant? Temperature? Light? So this will be our container. There's a little growth vessel here. I think it's at 200 milliliters. Perfect. So I've got graduated cylinders. We can measure out the amount of water that goes into here. So the amount of water will be the same to start with. The type of container will be the same across treatments. Little lids we can put on there, so the lid will keep them self-contained, keep them from drying out faster. And let the light in. And one of the things we want to standardize to begin with is the starting amount of the plant material. And that hadn't quite worked out yet. So these are basically our sorting containers. Take a handful of these and space them out. And I think one of the things we're going to have to do is to figure out how to standardize how much we have. So in my mind, they were, I think when I think about these, they're all the same size in my mind, but they have this kind of simplified here, sort of like a triangular growth off to the side. And you might have a few of those all still connected. So we could try, my initial thought was like try to get one of these each and try to get 10 of those in the container. So maybe back up a sec. How are we going to measure how well they grow? What do you think?
Take measures of them something like a week later.
Okay, what are we going to measure?
What do we measure?
Maybe. Actually, it might be possible. So we can kind of shoot for 10. I think they might come apart easily if you kind of agitate them. I'm not sure. You can shoot for pieces of the same size. Something like that, maybe. Let's see what I think that's a little more standard starting point. I don't know what it looks like to put 10 of those in. That might be a lot. There it is. Pen goes in there would be all right. So we could weigh them. The problem with weighing plants in the fresh state is that they're going to come along with some water. So it's a little hard to dry out a living plant and then weigh it as it's wet. It's not impossible. We could do that. I think maybe I'm hoping to see some fairly big differences. So what you could do is those little puppies on there, and we can take photos of them. And it's possible to use like an image analysis software, and you can just take the photo, maybe against a white background, and you can take this photo and standardize it to the square of what you see from directly above, and measure how many of the pixels in that photograph are black or green or whatever. So dark versus nothing. That would just measure the coverage. Okay. So we can actually watch them grow over time doing that. Let's plan for that. I guess I'm kind of working this out with you. I haven't done this before. So you've got certainly a lot of material to choose from. Along with the ferns are these little green circles. Those are actually a flowering plant called duck meal, or wolfia. And I'd like to try to separate those out if we can. So you might need a couple of washes with these. You transfer the fern and keep transferring until you don't have any of the duck meal left. Just as an extra variable, something we probably don't want in there. They might grow and take over. They might not have any effect at all. We'll just try to keep them out of there. I don't know. How's that sound for a thing to measure? So, no duck meal, just the Azolla. I think so. I put 2 of those size in here. I think if we got up to 10, that would be pretty reasonable to see the difference. A little bit of photographing, kind of going off to the side, but I think that's okay still. Let's take a photograph from top down, measure how much of the surface is open versus filled with plant. There. Okay, what do we record for the starting point? What do we want to investigate? How do we set up an experiment? I guess one last thing we didn't talk about is replication. So I don't remember how many of these I have. Got at least 50 of the growth chambers, and I can get more. So we have 4 treatments, 4 people in the room right now. What if we did 2 of each treatment? Out there. So if we do that, we got 4 in the room, 3 come to the next section, so we'll have about 15 replicates for each treatment. That's not too shabby. Okay, so water per vessel. So I didn't clean off the labels from the last experiment I did. You can just write with a Sharpie on the side if you want to just blot that out. Fresh Sharpie. Just put your initials on the side and then what the treatment is. And then if you have two replicates, you can call them A and B maybe, or 1 and 2. What kinds of treatments? I didn't talk about it, but we're going to put them into a growth chamber that has artificial light. And I can maybe figure it out as we go. We're going to randomize their order. So if we did this where we put all of the regular tap waters in a row, and then all of the tap plus nutrients in a row, if we put them in organized rows like that, we might have an effect about the position. So they go into a growth chamber and they have these linear strips of lights, and there might be different light hitting each of these rows. So if we give the different treatments different light, then we might see an effect that's based on the light that's not based on the nutrients. So if we randomize, that basically allows us to cancel that out. Or we could even keep track of their position and see if that has an effect later on. But I can do that as we go. We'll just use a computer-generated random number to assign where they're going to go. All right, I don't want to take up all of our time. How's that sound? Cool. Well, stop our recording. Hopefully you got something out of that.